Hello, everyone. My name is Injyil Gates. I am currently a senior at Howard University, studying maternal and child health with a minor in chem from Hollywood, Florida. And today I will be discussing single cell mapping of the small bowel and colon and focusing primarily on segmentation because that is what my internship was based on. So for the agenda, I'll have a brief overview over what the actual task was, where the collection sites were. I'll talk about CODEX multiplex imaging. I'll briefly cover image analysis and segmentation. I'll go over segmentation processes and the errors that occurred, and then I'll have my acknowledgement. So as a brief overview, I had the opportunity and the pleasure of working with the Nolan Lab at Stanford University. And we were tasked with mapping the complexity of the small bowel and the colon. And between the two organs, we had 8 collection sites in total. And you can see them here. I'm just gonna briefly discuss some of them. So like from beginning to end, you start with the duodenum. Right here, you have the duodenum, the mid-small bowel, and then the terminal ileum. You have the ascending, transverse, descending, and sigmoid colon. For CODEX multiplex imaging, this is the software that we use in order to process our data. It fulfilled the need for multi-marker spatial analysis, and it allowed us to see the spatial proximity of the cells within the specific tissues that we were looking at. The CODEX system uses 3 main parts, which is barcodes, antibodies, which are conjugated to those said barcodes, as well as reporters, which allow the fluorescent light to appear once the workflow process is complete. This workflow process is 3 steps: is stain, image, and analyze. And what happens is the tissue sample is stained with the entire antibody panel. And 3 at a time, the reporters are added so the fluorescent signal can be given if that marker is found within that tissue. Once this process is done, the next 3 is given, and then the images are layered on top of each other for analyzation. In order to view these images, so we have different ways. The first one is the composite image, which is basically allows us to view the fluorescent signal. And this data allows us to segment. The segmentation allows us to quantify the proteins that are expressed based on the amount of fluorescence that is there. And because of this, we're able to identify the cell types in order for clustering, as somebody mentioned earlier. And knowing these cell types can help us know the biology and understand it better. We are also able to locate the cell tissue for cell type mapping. And we're also able to see other cell types around this area for spatial analysis. Once the process is complete with the CODEX machine, we are presented with images such as these. Here we found 6 of the 44 markers that we wanted to locate in this specific sample, and they are shown in these different colors based on whichever marker they were. So again, just to reiterate, segmentation extracts the protein expression based on the amount of fluorescence in the cell, and this is what I focused on. So for the segmentation process, I would begin with an image such as this, where you can barely make out what it is. For those who don't know, it's cell nuclei, and I use a software called Annotator J coupled with Image J in order to get this picture right here on the right. So what this is showing, once we adjust the brightness and adjust the channels within that, we see in the green here, we have the cell nuclei, and in the blue, we have the cell membrane, and we're able to get a better view of this. So once we start segmenting, we are able to get a better outcome. So what segmentation is, before I go in and edit the segments, we receive a segmentation from a previous system, a computer algorithm that gives us these segments, and they're not always correct. So I will have to go in and edit these, and I will talk about the errors in the next slide, but that was what I was focused on for the duration of my internship. So some of the errors that came up was missing outlines, background noises would be outlined, and also some of the outlines for the cell nuclei will be combined as one. So here we see the missing outlines where they just completely fail to acknowledge the cell nuclei being present. We have background noise acknowledged, which is basically anything that's not important to the process. So it's not either a cell nuclei or a membrane, which I would have to go in and delete. And then the last thing is two cell nuclei being combined as one, and you can see it here and here. And this is just due to the closeness of the proximity of the cells, or the algorithm failed to realize it was two cells. So once we go in and fix an entire image, and these images can take anywhere between 3 hours and 8 hours, we produce images like this. And the purpose of this segmentation process is so that the algorithm can give better segmentations to future data sets. So eventually, we don't have to go in and edit these cells or fix corrections that need to be made. So I'll briefly talk about what I learned. I got a better understanding of the small bowel and the colon anatomy. I was able to understand the basics of CODEX multiplex imaging. I had a better understanding of cell segmentation. I learned how to navigate annotator J and image J, and I had the opportunity of speaking with a lot of people with career paths and personal testimonies. And for my acknowledgements, I would like to thank HuBMAP, NIH, specifically Zarina, AJ, Julie, for giving us all the presenters and the guidance. I would like to thank the Nolan Lab, Gary Nolan specifically, for allowing me the opportunity to just work in the space and be there. I would like to thank John Hickey for guiding me and helping me with this presentation, because without him, it would have not been done. I would like to thank Gulan for providing me with these segmentations. Shakira for helping me learn my way around the lab. Even though it was virtual, she still gave me that one-on-one experience. And everybody here is the people that I met and gave me personal testimonies about not only this internship, but about my career. And I really appreciate that. And lastly, I would like to thank all of my peers who completed this internship with me, because it was not easy at all. And thank you everybody for listening.